All right, hello. Let me just check if the mic works. One moment. All right, hello. Let me just check if the mic works. One moment. Right, hello. Okay, yep, it works. Okay, let's start. Cell junction and cell polarity. Hmm. So, what is cell polarity? Well, it is the spatial differences in the shape, structure. Wait, what is happening? There. 1 second. Yep, there we go. So, cell polarity refers. It is the spatial differences in the shape, structure, and function of the cell. The cell polarity is the spatial differences between the shape, structure, and function of the cells. Almost all cell types exhibit some sort of cell polarity, which will enable them to carry out specialized cell functions. Okay, the cell polarity is the spatial differences in the shape, structure, and function of the cells, and almost all cell types exhibit some form of it. You have the apical or apical surface above and the basal surface. So the apical surface, it faces the external environment or the lumen. Okay? And it's for absorption like microvilli, secretion or exocytosis, and protection. You have the cilia or mucus production over there. Meanwhile, the basal surface would anchor the cells to the basal lamina, and that would interact with the connective tissues. All right? So the thing is, it contains integrins which will make it adhere to the ECM, which is the extracellular matrix. So again, what's the difference between the apical surface and the basal surface? Apical faces the external environment. Meanwhile, the basal surface would anchor it to the basal lamina. You have a couple more things there. You have the epithelial sheet, basal lamina, and connective tissues. The epithelial sheet would form a continuous barrier which would separate the different environments, and cells are tightly packed and polarized with distinct apical and basal surfaces. So you have the sheet, and basal lamina is just a specialized ECM layer which would support the epithelial cells. So the basal lamina has collagen, laminins, and glycosaminoglycans. Okay. And it provides structural support. So the basal lamina has the collagen. There's also the connective tissues, which is beneath the basal lamina. So what's the order? You have the apical surface, epithelial sheet, basal surface, and basal lamina. The connective tissues below it would provide mechanical support and nutrient exchange. It has the macrophages, which is for defense, which is for immune defense, I mean. And then fibroblasts would produce the extracellular matrix proteins, such as the collagen and elastin. Mast cells would be for immune response and inflammation. Meanwhile, the capillaries is for nutrient and gas exchange. So the connective tissues has different stuff that has collagen, macrophage, fibroblasts, which will produce the collagen and other like elastin. And then you have the capillaries, which would be for gas exchange. Why is cell polarity important? It's important because it regulates the directional transport, and it maintains tissue integrity and facilitates signaling and interactions with the environment. So again, why is cell polarity important? The basal surface, it would regulate the directional transport regulates the transport, maintains the tissue integrity. So it's useful for transport. It maintains the tissue integrity and facilitates the interactions for signaling. So it's signaling, transport, and integrity. TIS. Transport, integrity, and signaling. So, and also it's essential for organ function like the intestines, kidneys, lungs, and the skin. For example, for hepatocyte polarity, you have the endocrine function, which is like blood facing surface or the sinusoidal domain, and that will interact with the blood, exchange of nutrients. You also have the cell junctions, the tight junctions, desmosomes, and gap junctions. So the tight junctions, this one is the occluding junctions. It maintains the compartmentalization by sealing the paracellular surface. So it would, tight junction is for compartmentalization, desmosomes is for mechanical strength, gap junction is for communication. So you have three so far: tight, desmosomes, and gap. Tight junction would be for compartmentalization. Desmosomes, how can I make sure? Desmosomes, mechanical strength. Desmo strength, okay. Gap junctions is for communication, which would allow small molecules to pass between the hepatocytes. So again, tight junctions, desmosomes, and gap junctions. Tight junctions would be for compartmentalization. Desmosomes is for mechanical strength. What is the third one? Gap junction is for communications. And then you have exocrine function, which would secrete, it's the bile facing surface. So again, we are talking about the hepatocyte. You have the endocrine function, which is the blood facing surface, and exocrine function, which is the bile facing surface. So secretes the bile into the canaliculus, which falls towards the ducts. Bile production. Okay. So you have, this is, okay. Hepatocytes play a crucial role in biosynthesis and secretion. And you have a couple of transporters and functions, okay? This might not be in any particular order. So first, you have the uptake from the blood, which is the NTCP, OAPS, and OST. Sodium co-transporting polypeptide uptakes the bile salts. And essentially, the ATP-dependent transporters help remove waste products and regulate bile composition. The bile canaliculus. What is the bile canaliculus? Good question. Bile canaliculus. It is the smallest bile duct between the hepatocytes. So the bile canaliculus is the smallest bile duct in the hepatocytes, and it is sealed by tight junctions, preventing the bile leakage into the blood. And also, it has dense microvilli lining the canaliculus, which increases the surface area. The bile canaliculus is the smallest bile duct between the adjacent hepatocytes, and it's sealed by tight junctions, preventing bile leakage into the blood. And the dense microvilli would line it. Smallest bile duct, and it is lined by microvilli. Also has tight junctions, so there's no leakage. Again, remember, tight junctions is for compartmentalization. Just for compartmentalization. Let me just get the sticky notes. Sticky note. Note. Next. Okay, and we're gonna talk about enterocyte polarity and macrophage polarity now. So enterocyte, that's in the intestines, and it is the absorptive cells of the small intestine. Also has apical and basal polarity for nutrient uptake, and the apical surface features microvilli for maximized absorption, while the basal surface would anchor it to the basal lamina. Nutrient transport for circulation. Tight junctions again maintain the barrier integrity and prevents leakage. Okay. Macrophage polarity, meanwhile, unlike epithelial cells, macrophages display functional polarity. Functional polarity, that's different from the others. It would adapt its shape based on environmental signals. Macrophages have pseudopodia for movement and phagocytosis, which would respond to infections by polarizing into pro-inflammatory M1 or anti-inflammatory M2 states. So, macrophage polarity it would display functional polarity. It has pseudopodia and that would just crawl for movement and phagocytosis. It would respond to infections, either M1 or M2, M1 being pro-inflammatory or M2 anti-inflammatory. That's a new thing. I did not read before. Let me just mark it. And this flexibility would allow macrophages to balance immune defense, tissue repair, and etc. Again, what's a cell function? It is, a cell junction. What is a cell junction? It is an intercellular bridge, a type of structure that exists within the tissue of some multicellular organisms. Cell junctions consist of different multi-protein complexes which provide contact between the neighboring cells. So cell junctions is a kind of a bridge within cells and would provide, it has different proteins and which would provide contact between neighboring cells or within that cell through its extracellular matrix. Some important things would be tight junction, adhesion belt, desmosome, gap junction, integrin, selectin, focal adhesion, hemidesmosome, and etc. Okay, so we have two things. We have the cell to cell and cell to membrane. Sorry, one more. Cell to cell junctions and cell to basement membrane. So cell to cell junctions would be earlier the tight junction, adhering junction, desmosomes, and gap junctions. So tight junction would be zonula occludens, meanwhile adhering junctions, zonula adherens. So tight junction, occluding junctions would be, it is associated with areas of two cells where the membranes join, forming a virtually impermeable barrier to fluid. So it would really, really regulate the passage of ions, water, and others. And if you whack that up, that is where edema, jaundice, diarrhea, and bloodborne metastasis. Okay. So tight junctions, it is occluding or zonula occludence, and it would really regulate the fluid or passage of ions. Adhering junctions, meanwhile, would be a bit more basal compared to tight junctions, and the adhering junctions or adherence junctions would lead to the formation of tight junctions. That's important. Formation of adherence junctions leads to the assembly of the tight junctions. Desmosomes is for mechanical strength. That's macula adherence. It is for It is arranged on lateral sides and would resist shearing forces. And it is found in simple and stratified squamous epithelium. So cell-to-cell junctions. So far, we have talked about tight junctions, adherence junctions, desmosomes, and then now you have gap junctions. Gap junctions would also be for passages of ions, and it is needed for electrical coupling. It's for signaling, the talking. That's why there's a mini gap. And then you have the cell to basement membrane, which is the hemidesmosome and the focal adhesion. Hemidesmosomes are very small structures on the inner basal surface. And while desmosomes link two cells together, as mentioned earlier, for the shearing forces, hemidesmosomes attach the cell to the extracellular matrix. Okay? It anchors the epithelial cells to the basement membrane. And then you have, so again, that's the difference between the desmosomes and hemidesmosomes. Desmosomes is cell to cell. Hemidesmosomes attach the cell to the basement membrane. And then you have focal adhesions or actin filaments. It's also called cell matrix adhesions or FAs. And they are large macromolecular assemblies which mechanical force and regulatory signals are transmitted between the ECM and the cell. So it anchors the cells and transmit the signals, which facilitating the environmental response. So again, what's the purpose of focal adhesions? It would be for mechanical forces and signals are transmitted between the ECM and the cell. So tight junctions again. Let's go back. Cell-to-cell junctions, you have had tight junctions or zonular occludence, adherence junctions or zonular adherence, desmosomes, macular adherence, gap junctions. Tight junctions would be to really control the passage of fluid and ions, and if there is leakage there, then that's when you have edema, diarrhea, and bloodborne metastasis, and maybe jaundice. Adherence junctions would lead to the formation of tight junctions. Desmosomes would connect the cell to cell as well, cell-to-cell adhesion, and would resist the shearing forces. It is for the mechanical forces. Gap junctions would be for communication and signals. Cell to membrane is hemidesmosomes, and then you have focal adhesions as well. Mechanical force, and it's also for talking. Just digging a bit more on for cell junctions. Again, you have tight junctions. In distinct plasma membrane domains, it is for diffusion barriers, prevent lipid and protein mixing, and really regulate paracellular support for selective ions and molecules to pass while restricting leakage. And the important parts is okay, look at this. You have ZO1, ZO2, ZO3, and JAM1. Those are proteins or markers to check for tight junctions. I will write that down again. Euclidean tight junctions. You have markers such as ZO1. Okay, so technique. If you have freeze fracture electron microscopy, which would reveal tight junctions as ridges and grooves. So electron microscopy images could reveal the tight junction strands as different ridges and grooves, and these show how the junctions create the distinct apical and basolateral membrane domains. Some important proteins for it again is claudins, occludin, and junctional adhesion molecules, and claudins are primary sealing proteins. Occludin would support the barrier function and interacts with ZO1. So claudins are primary sealing proteins. Occludins would support the barrier along with ZO1. ZO1 links tight junctions to actin skeleton. So those are very specific proteins for the cell junctions. Claudins sealing, occludins for barrier function. ZO1 would link the tight junction to the actin skeleton. Junctional adhesion molecules. Junctional adhesion molecules. They are members of the immunoglobulin superfamily, and it assists in cell adhesion and signaling. Okay. Adherence. It initiates the cell-to-cell contact and forms tight junction. Cadherin is for adherent. Adherent. Sounds alike. Let me adherent. Cadherin is a calcium-dependent adhesion protein that facilitates homotypic interactions between the cells. And it links to the cytoskeleton via. So how is cadherin links to the cytoskeleton via the catenins alpha, beta, and gamma. And it is a transcription factor that plays a role in the Wnt pathway. I think we will talk about the Wnt pathway later. So again, cadherin is an important protein in adherence junctions, and cadherin links to the cytoskeleton via alpha, beta, and gamma catenin, and beta catenin is important for Wnt pathway. Cadherin focuses on, well, we have the structure of cadherin and the calcium-dependent adhesion of it. Oh, we're here. In adherence junctions, the beta catenin would bind to cadherin and alpha catenin, I mean. So in the adherence junctions, beta catenin binds to cadherins and alpha catenin, which would link to the actin. And in the Wnt signaling pathway, beta catenin is stabilized in the presence of Wnt, and then it would enter the nucleus and regulate the transcription of genes like cyclin D1 and c-MYC for cell proliferation. This is a very important detail, especially later on. I have to use a different tab. So again, in the Wnt pathway, beta catenin would stabilize the presence of so if you have Wnt, it would stabilize beta catenin. Beta catenin will then enter the nucleus, and then genes of c-MYC and cyclin D1 will be transcribed. Those are crucial genes because they influence cell proliferation. Got it. And without the Wnt signaling, beta catenin is degraded by a complex involving APC and axin and GSK3 beta. So the important detail would be the fact that wait 1 second, there you go. The important detail would be the fact that You have different images. Okay? It connects beta catenin. Okay, there you go. You have the Wnt signaling. If you have Wnt, it stabilizes beta catenin. Beta catenin enters the nucleus. It regulates the transcription of genes such as cyclin D1 and c-Myc. And then after that happens, cell proliferation is increased. And if there's no Wnt, then beta catenin is just degraded by APC. What are some evidence for losing cell polarity? Okay, they knocked out genes. Well, the scientists would knock out genes which are involved in membrane lipid synthesis to see how it would affect polarity. So how to check? You knock out the specific genes involved in membrane lipid synthesis. If you knock out the genes involved in membrane lipid synthesis, you might be able to see if polarity is affected. And some scientists did that with liver cells, because if a cell loses polarity in the liver cells, then you have bile leakage, and then the plasma would turn yellow and jaundice. That's essentially it. And what they knocked out was the SPTLC gene, SPTLC gene, and it's connected to the beta catenin pathway. Okay? Normally, beta catenin remains bound to E-cadherin at the adherence junctions. The beta catenin normally is attached to E-cadherin, and then the plasma membrane sphingomyelin levels are normal. But in the SPTLC2 knockout, there is reduced for the plasma membrane sphingomyelin. So then, if you knock out the SPTLC2, it lessens the attachment, and you have weaker adherence junctions now. And then you have leakage. Beta catenin detaches, translocates to the nucleus, and then you have weird transcription, thus tumorigenesis. And it's very clear because there is bile leakage there, causing yellow plasma, aka bilirubin accumulation. And that means SPTLC2 deletion disrupts the beta catenin pathway, which is linked to membrane polarity and tumor formation. Okay. Green sticky note. Experiment. And then you have the structural and ultrastructural evidence of polarity loss. It's just showing the different wild type and SPTLC2 KO liver cells. That's fine. So just to sum it up, cell polarity loss in knocked out cells would lead to disruption of bile transporters. The jaundice in mice indicates that there is defective bile secretion, and the liver hypertrophy and tumorigenesis occurred in the KO knockout models and cancer. Because remember earlier, the Wnt signaling, if you have Wnt, then beta catenin can go towards the transcription and c-Myc is formed, and if that is really, really disturbed or increased, you might have too much cells then. So some steps. Okay, to show it, you could do immunofluorescence staining for the cell markers. You have specific antibodies for the different liver tissue. And then you can do a bilirubin assay. Bilirubin assay will just show the bilirubin levels. Serum from wild type mice would be clear. And then KO mice would show yellow tints. EM could show the canaliculi, while KO cells would have vacuoles. The vacuoles of it would have disrupted canaliculi. Histology, well organized for wild type, KO would have very disorganized architecture. So and then liver weight, it's gonna be enlarged for the KO. So serum, bilirubin, immunofluorescence, electron microscopy, and histology. Immunofluorescence for the antibodies. Serum bilirubin would show the differences between the wild type and KO. Electron microscopy could show the structure of their canaliculi, while histology would show the disorganized architecture. Desmosomes was for strength and shearing forces, and it provides mechanical strength to tissues. Structure, it has adherents such as desmoglein, and desmocolin. It spans the intercellular space and mediates adhesion between adjacent cells. Okay. Desmocolin, desmocolin. In the picture, it forms extracellular adhesive interface. Just remember desmoglein and desmocolin for desmosome. And I will also write that out somewhere over here. Where was it? Yeah, beta catenin was for adherence junctions. And Wnt. Desmosome is adherent also, but it is desmoglein and desmocolin. Desmocolin. Ooh, I think we can add, what was it? It is, no, it is stream buddies, I think. This one. There you go. Yeah, that one. Okay, that's good. We can add that. Next. The second image would, yeah, we did that one already. Desmosomes, desmoglein, desmocolin. We also have the desmoplakin, placogoblin. Specific terms. How can we study desmosomes? Okay. Interesting question. To study it, you can do a lot of things. So, let's see. You have immunofluorescence microscopy. Why? Because if you do that, well, first, before you actually check them out using immunofluorescence, you have to stain them. In wild type cells, the desmoproteins are localized at cell junctions, right? While in mutant, there might be loss of these proteins. So, if you check or stain for it, fixed tissues or cultured cells could be permeabilized. Well, that would help you check it, right? I had an extra sheet of paper here. Did I lose it? Oh my goodness. It is lost. Paper. Oh, there it is. I just have to list it out. Immunofluorescence. This is from Cell Junction. Immunofluorescence microscopy. Mm, there was also the freeze something. Freeze fracture. And no. No, not that one. Where? Nope. Wrong page. Right. Oh, there it is. Found it. Freeze fracture electron microscopy. Freeze fracture electron microscopy. But essentially, the electron microscopy would show the desmosome structure. Okay, Western blotting for the proteins. You will see the expression levels. Western blotting. Page 3. And then you have the functional assays for the strength of desmosome mediated adhesion. Just to check co-immunoprecipitation also. So, okay, let's go from the beginning of this one. To analyze desmosomes and their role in the protein adhesion, you can do a couple of things. Immunofluorescence would check for the desmosomal proteins. You can fix the tissues or cultured cells and incubate them with primary antibodies and then fluorescent antibodies, the secondary ones. And then confocal, you use confocal to check. Wild type would have it. The knockouts might not have it, or it is all over the place. Western blotting of the protein for the desmosomal proteins, you might confirm the expression levels of desmoglycan, desmocolin, and etc. First, you separate the protein extracts using SDS page, because with that only the size is the important detail. The charges would be removed because the SDS would coat it all in the negative charge, so the charge isn't the it's not the main character. Okay, next, electron microscopy would show the different structure. You fix and then you dehydrate and you embed it in the resin, and then ultrathin slices, you stain it, and then you look at it using transmission electron microscopy. Co-immunoprecipitation, that would be to check for the interaction. It's co-immunoprecipitation. So the cell lysates you incubate with an antibody against a target protein, and the beads are used to pull down the protein complexes. After the washing, you check via Western blot, and that will check the binding partners. Wild type samples are expected to show interactions between the desmosomal proteins, while mutant would have this weakened or absent interaction. Next to gap junctions, that is for when the cells are talking to each other. Passage of cells, electrical impulses and ions. They have channels such as connection, but it's a weird spelling, CONNEXONS, which is for cell communication. Write that up. Take notes. Where is my thing for connect? CONNEXONS. Connections. Google connections or connections. Connections or connections. Oh, those are different. A connection is composed of 6 transmembrane proteins called connexins. Okay, write that out. Connections are composed of 6 transmembrane proteins called connexins. 6 of them. So we have an electron micrograph of a gap junction with how different connections. How can I know? O and I. I is inside. Okay. Oh, that's funny. It does look like. Okay, it has. It really has. Connection A, connection B. Just loops. It's open and closed. It can have open and closed states. Each connection is consists of 6 connection subunits with 4 transmembrane domains. 1, 2, 3, and 4. It has loops. Different loops. Oh my goodness. So it's like a flower. If connection is the flower, then the petals are connexins. You have 6 petals. 2, 3, 4. No. 1, 2, 3, 4, 5, 6. And then that's like a tall one. And that whole thing is like an M1. That's the first domain. And then it loops up and down. M2, M3, M4. That's the 4 transmembrane domains. Okay? And then there is E1 and E2 for the loop. The extracellular loops, I mean. But you have the cytoplasmic loop, the CL, at the bottom. And that would regulate pore permeability, voltage gating, and etc. Crucial detail. Next. Experimental methods to study the gap junctions. Okay, that's similar. You could do immunofluorescence for the proteins, Western blot, confirm the expression. You have a dye transfer assay. You have to also be green. Dye transfer assay for gap junction communication. Why? Well, it would check whether the gap junctions allow the transfer of small molecules between the cells. Donor cells are loaded with a fluorescent dye, such as lucifer yellow, and those can pass through the gap junction. After the incubation period, fluorescence would spread to the adjacent cells, and that's observed using fluorescence microscopy. In wild type, extensive gap transfer. Meanwhile, in extended dye transfer, in the gap junction deficient cells, almost no, it won't transfer the immunofluorescence. That means intercellular communication has been screwed up. All right, and dye transfer assay. Next, you have dual Hall cell patch clamp for electrical coupling. That is when you check its conductance. And in wild type cells, the gap junction would exhibit the conductance while knockout would not. Fluorescence recovery after photobleaching. We said that in the cell membrane discussion, where you photobleach all of them, and then the cells expressing the fluorescently tagged connexins would hopefully talk to each other and would pass it on, and there's supposed to be fluorescence recovery over time. The knockout ones would not have, or would have delayed or absence recovery, so that's how you track the gap junction trafficking. Co-immunoprecipitation would just check the interactions between the proteins. Okay, I have to double check. Versus Western blot. Because I think co-immuno would just check. Oh yeah, it's similar. Study it. Cell lysates are incubated with an antibody targeting the connexins, and the beads are used to pull down the protein complex. And after washing, the bound proteins are analyzed by a Western blot. In the wild type, you expect the binding partners 0-1, the binding partners of connexins, while the mutants you cannot see. The cell to membrane junctions, you have hemidesmosomes again. It connects cells to membrane. ECM, you have different integrins. Integrin family, such as laminin and collagen. Okay? Integrin family of transmembrane proteins play a key role in hemidesmosome function by linking the intracellular keratin filaments to the ECM protein. I will just have to quickly write that out. Where is my mini note? Integrin, and that is also laminin, keratin, laminin, collagen. Collagen. Alright. Next to, oh, we have a disease this time for the page. You have the bullous pemphigus. I might be butchering. It's a skin blistering disease. It's an autoimmune disease. And it is caused by autoantibodies such as the BP180 and BP230, which disrupts the hemidesmosomes and leading to subepidermal blister formation. So if you whack up the hemidesmosomes, aka the cell to basement membrane, you would get subepidermal blister formation. And the normal skin usually is the epidermis is anchored to the dermis via hemidesmosomes, okay? That's the crucial line that we have to remember. Epidermis is anchored to the dermis via hemidesmosomes. And if you check it via immunofluorescence staining, you can, using the BP180 antibody, you could see it. Okay? Meanwhile, if you have signs of the bullous pemphigoid, then there is tense blisters, and there is epidermal detachment and inflammation. So normally the epidermis is connected to the dermis via hemidesmosomes. And if you want to check what is going on in there, you can check via immunofluorescence staining using BP180 or BP230 or collagen. And loss of it would lead to possible detachment, epidermal detachment. Alright? Alright. Here. Focal adhesion. That is also called cell matrix adhesions, focal adhesions. Integrins serve as key adhesion receptors, which connects the extracellular matrix to the actin skeleton. You have integrins, actinate, heterodynamic proteins with different subunits. And the function of integrins would be for cell survival. It's just large macromolecular assemblies which would relay mechanical force and signals. That's the crucial part about focal adhesions. So again, that is focal adhesions. All right, this is a mini review of both the cell membrane and what was it? The cell membrane plus cell junctions. Why do we need cell signaling? Well, cell signaling would be where the cells communicate with each other, and that would contribute to growth, differentiation, immune response, and tissue homeostasis. And the signaling would help with apical and basal polarity. What kind of junctions exist and what are some examples? You have the tight junction, adhesion junction, desmosomes, hemidesmosomes, and focal adhesions, cell-to-cell interactions, and membrane-associated junctions. We have ecadherent claudins connections. No, connections are composed of proteins, subunits such as connexins. And to check remember, how can I determine if GEM1 is in the lipid raft region? I could use it was the detergent one, cold detergent raft. I could use Triton X at 4 degrees Celsius, the C, not the F. Triton X. I could use Triton X for that one. Okay. For Triton X. Okay, you have both. But lipid rafts would not solubilize, okay? So you first lyse the cells in the Triton X, then ultracentrifuge it. You fractionate the lysate, and then analyze each fraction by a Western blotting. And you would expect that the GEM1 is in the lipid rafts, aka in the low soluble ones at the top one. Non-raft would be in the soluble region. And if you would really, really like to check it, you could use confocal microscopy with cholera toxin, aka GM1 marker. That one's done. Does adherence junction formation determine tight junction formation? Yes, because, well, how do I check that? Well, to test it, you could do an ecadherent knockout. Because if you knock out, you can use CRISPR or siRNA to knock out ecadherent. And if you do that, if adherence junctions are really important for tight junctions, you would expect that there is no formation of tight junctions. So you can check tight junction formation using Western blot for tight junction markers, such as the Z01 claudin occludin. And you can also use functional assays for the permeability. And if tight junctions do not form, you can do a rescue experiment. So first you knock it out. You knock out ecadherent using CRISPR or siRNA. And you assess the tight junction formation using Western blot. Check for Z01 markers, Z01 proteins. And you rescue it by transfect the KO cells, the knockout cells, with a wild type ecadherent. And if the tight junctions are restored, then voila, you have it. Okay, you have bacteria in the GI tract. So here's an experiment. Bacteria in the GI tract can produce lipopolysaccharides, which under normal conditions do not enter the bloodstream due to the presence of tight junctions in intestinal epithelial cells. Hell, I need to drink water. So bacteria in the gastrointestinal tract can produce lipopolysaccharides, which normally do not enter the bloodstream, okay, because of tight junctions. But if the tight junctions are compromised, the lipopolysaccharides can leak into the bloodstream. which would lead to severe systemic effects. Okay? And that is bad. And you can also check for that. How can you check it? Well, you generate a knockout model. Whoops. 1 second. You just generate a knockout model, alright? How? Using CRISPR. Using CRISPR, you knock out the target gene in the mice, and then you validate the knockout at the genomic level, like PCR. Also you can check the protein level using Western blot. And after that, literally to assess the intestinal barrier function, you can use the LPS blood test. Because if the tight junctions are compromised, the gut bacteria should leak into the bloodstream. And how can you do that? Well, you can use an assay or ELISA. Assay. ELISA. This is for next. You can also use a blue dextrin permeability test. How does that work? Well, you can feed the mice the blue dextrin. And after a set of time, you can collect blood samples from them. And if you see high dextrin levels in the blood, that would mean that there was increased gut permeability, which is bad because that means the tight junctions was whacked up or compromised. You can also image the gut integrity of the mice using fluorescent tracers to visualize the barrier integrity in intestinal tissue sections, okay? And then you can do a rescue experiment, which would reintroduce the wild-type gene via adenovirus-associated vector. Reintroduce the gene using an adenovirus. How can I remove the gene in CHO and HO 7 cell lines? To knock it out, I can use siRNA, shRNA, CRISPR, and that's CRISPR-Cas9. So siRNA, that will, let me just write it, that's a good one. RNA or CRISPR-Cas9. siRNA is it's a temporary knockdown. shRNA is stable knockdown. Meanwhile, CRISPR is permanent. Again, siRNA would bind to the target mRNA, leading to mRNA degradation via the different via RNA-induced silencing complex. And then you could check the knockdown via qPCR because you can check the mRNA levels, and Western blot would check the protein levels. Short hairpin RNA, that's stable because short hairpin RNA is transcribed from a vector and then forming the hairpin RNA that would target the mRNA again. And then check it via different stuff. It's not a full gene knockout, but it can be it can go into the genome. It can be integrated into the genome. You know, CRISPR is really, you introduce a double-stranded break in the DNA, and it's repaired by the non-homologous NHEJ, which would cause gene inactivation. And yeah, you can check via PCR again and Western blot. Oh, where is it? How would you determine if a junction gene is upregulated? Well, you can do both Western blot and mRNA qPCR. Because Western blot would confirm the protein upregulation. You extract the protein, SDS page, transfer to the membrane, incubate primary and secondary antibody, and then you detect via chemiluminescence. If it is really upregulated, the band intensity will be stronger. qPCR would confirm the mRNA upregulation with its values. The qPCR would have the primers for the junction protein. And if it was upregulated, the gene expression or CT value will be higher. And then you also have the post-translational modification check. It would use the antibody in Western blot to check if it was phosphorylated. How can I determine the size of 3 different proteins? Good one. Well, for that, where was it? Sorry, I lost the page. SDS page or size exclusion. The SDS page, well, you treat them with SDS to give them uniform negative charge, so they flow through. You load it on a polyacrylamide gel. Smaller proteins are faster, large proteins are slower. So there is no charge influence, and only the size is the determinant of the migration speed. Size exclusion. Larger proteins elute first, smaller proteins get trapped. So that's how you can determine the sizes of different proteins. You know, RNA sequencing, how does it work, and what are its pros and cons? You extract the total RNA, and you remove the ribosomal RNA, convert it into cDNA, and use next generation sequencing. The map would read the genome. So this one is interesting. It provides a global transcriptomic profile and which genes are upregulated or downregulated, but it can't show you too much because you extract the total, remove the rRNA, convert the fragmented RNA into cDNA, and use NGS, next generation sequencing, to sequence the cDNA, the whole genome. And how can you validate the RNA-seq results? Well, you can use qPCR again, and Western blot and functional assays. All right. And I think that is all for that one. Thank you for watching. Bye.